So I thought I'd start today with a little background in some PowerPoint slides regarding one of our new workflows, which allows us to achieve highly accurate variant calling with NGS data. And more importantly, we provide means to the customers to determine the accuracy of their own, essentially their own workflow. And so we built that into our software so that when you get your own data, you can run an assembly, do the variant calls, and our software will calculate the accuracy for your data sets, which is a really important piece. Rather than just looking at the marketing information that I'll give you today where we ran the assemblies, I think it's really important that we put that ability in the customer's hands so they can troubleshoot their own process and have a good feel for exactly how accurate their own process is. And so we'll spend some time discussing how that's done, different points in the process where you lose accuracy and how you can assess where you've lost that accuracy. We provide nice visualizations so you can go and look at those places in an assembly, run the appropriate reports, and then kind of the final piece of that is a calculated accuracy at the end, and that's literally assessed by determining how many variants you can detect in a known sample, right? So if you've got 10,000 known variants in a sample, in a control sample, do you see all 10,000 in the end report, or do you have 9,995? And if you have less than 10,000, where did those 5? Why did you miss those 5? And you can interrogate that. So those are the kind of things that we're going to discuss today. As a starting point, though, I'll give some background to DNASTAR as a company, and then we'll go into kind of what the workflow is, and then we'll launch the software, and I'll show you how to set up one of the projects and do some of the downstream analysis. So DNASTAR is located in Madison, Wisconsin. It doesn't look quite like that now. We're in the middle of fall, so I see lots of nice fall colors out my window right now, and snow will be coming soon enough, but it looks pretty nice like this right now. DNASTAR is compatible with all the NGS platforms. So what we'll be talking about today is mostly Illumina. Illumina has really set the benchmark for accuracy. And so to get the most accurate assembly and analysis, we'll choose Illumina data to do that. Ion Torrent can also be accurate in certain instances and circumstances. And so we do have some white papers. I'm not going to discuss them today in detail, but we do have information about Ion Torrent accuracy and how to best work with that data. 454 is kind of on the way out, but PacBio is used more for de novo assemblies and some other kind of workflows. And of course, we're looking ahead to nanopore technologies to be compatible with them as well. The hardware that we're talking about to do these assemblies and downstream analysis is modest hardware. We've designed the algorithms to be optimal on desktop computers. In many cases, laptops are sufficient. And kind of a basic setup for doing alignments. If you're working in a human system and you want to align data to, say, an exome or a gene panel has gene represented on all the different chromosomes. A computer to do that sort of work could be a sturdy laptop with 16 gigs of RAM. The important part is to have enough disk space. A lot of the processing with the algorithm is done on disk. So I always recommend a dedicated hard disk. And it can be an SSD drive, it can be an internal or external drive, but for human exomes, about a 1 terabyte disk is sufficient. And as long as you have that disk, most of the newer laptops now, you can run exome assemblies. It might take a little longer than a big desktop computer, but we're talking, maybe 3 hours versus 2 hours. So we get really good, fast performance on relatively modest and inexpensive computers. And if you have questions on hardware, I'd be more than happy to answer any of those kind of questions that you have. So another option for folks is not to use local hardware, but to use the hardware up in the Amazon cloud. And we see there's a trend more and more that, and it comes from all different areas, but in many cases it's IT folks that say it's too expensive to maintain and replace computers every 3 to 5 years. It's actually cheaper in the long run just to use cloud computing. And Amazon, of course, has very nice, shiny new hardware up in their cloud. So you get top performance when you use the cloud. And the way that we typically use the cloud from DNASTAR is that we set the assembly up on our local computer, and we just make the cloud connection, and it uses a secure HTTPS connection to upload the data, and then the data is assembled up on the cloud computer, and then you can return it down locally for analysis. And there's some advantages to that. One, you don't have to store all the data locally. Two, you don't have to maintain hardware. You can have very minimal hardware. That's LAN's hardware. And then another big advantage is that you can run multiple assemblies concurrently. So, for example, if I've got 100 human exomes that I need assembled in a day, I can send the job up to the cloud and, within a few hours have all the results returned to me on my local computer. So, again, it's more, looking to the future, more and more folks are shifting to cloud computing, and this is a really nice way to use cloud computing. Our software also comes with support, so it's a big part of commercial software is that it's fully supported by a whole team of people here. I do a lot of the NGS support. We have other folks that can answer some of the NGS questions. I have access to the developers that wrote the software, so if we have advanced questions, I can meet with them and we can figure out more complicated problems and get back to the customer in a reasonable amount of time. We also have videos that are anywhere from 1 to 8 minutes long on our website, and these are on specific topics. So if you're doing a certain thing and you want to see how to do that in our software, you can find this again under our support section. We also do webinars like this about once a month with different people. Again, my webinars are typically focused on NGS, but we have other webinars on things like structural biology and different molecular biology tools as well that are available in our suite. So if we don't cover things today that you're hoping that we could cover, certainly look at the archived webinars that we have, and we may have covered that topic recently. So one of the first questions that comes up with using software on a local computer and not, say, on a big Linux cluster is, are you going to be sacrificing speed? If I use some open source software up on a cluster, is that going to be a lot faster than running commercial software on my laptop or on my desktop computer? So I like to show these kind of benchmarks just to give you a feel for how fast these algorithms are. So for example, the typical benchmark is a human genome data set, with Illumina data, and those are typically, about 1 billion Illumina reads, 100 base pair Illumina reads, typical human set, and that gives you close to 40x coverage. Our assembler can take all the data, assemble it or align it to the human genome, do all the statistical Snip calling, and build all the output files, the BAM files, all the Snip tables, all the coverage tables, and do all that building in about 13 hours. So it's a very fast processing. A lot of the open source programs will take more than 13 hours just to do the assembly, and then you have to go and build one chromosome at a time, go through and build all the variant files, and then figure out how to do the analysis. So it can be a big, big time saver having a piece of software that can just kind of go from A to Z in a short amount of time. Exomes, more folks are actually doing exomes right now than whole human genomes. A couple of hours for an exome is certainly reasonable. Again, it depends on the coverage. 50x is common, but if you have a deeper coverage exome in the hundreds, it'll take a little bit longer the deeper the data sets get. Smaller projects like cancer panels or E. coli genomes, we typically measure those in just minutes. Right? So it's a really fast algorithm. Our focus initially was speed. So when we first developed these algorithms several years ago, we wanted to be the fastest thing out there. We want to be able to crunch through a lot of data. And then really in the last 2 years, the focus has really shifted to, let's be the most accurate aligner and SNP caller that's available. And so really for the last, a little over 2 years now, we shifted that focus. And a big part of the reason for that is that a data set, a highly validated data set, was made available through the NIST at the NIH, and it's from the Genome in a Bottle Consortium. And this is human genome. You'll hear me refer to this again, but it's NA12878. It's the most sequenced human genome that's out there. And so because of all the data from from Illumina and Ion Torrent and 454 and Solid, the variants then were verified, many of them through Sanger sequencing as well. So there are millions of variants for this genome that have been verified through multiple methods. So we're very confident where the variations are in this data set. So it's the perfect data set to use to calibrate software. And so once we had access to that genome in a bottle data, we're able to really fine-tune all the different steps, the assembly process, the trimming, the Snip calling, the Snip reporting, the Snip filtering. You can always check it against that answer. A very large answer that has, in an Exomes case, 15,000 data points in it. And those are really the kind of data sets that we needed to improve the software and make it as accurate as it is now. So right now our accuracy, and we'll look at this in more detail, but our accuracy with Illumina data on these Exomes is about 99.7. The genome in a bottle answer isn't 100% accurate. It's close. It's probably 99.9 or 99.95, somewhere in that range. So we're at a point of just a very small percent of error that we continue to investigate. We look to see, can we improve anything to pick up these last few variants that we're missing? Or in some cases, we find that there's an error in the answer, and we go back to the Genome in a Bottle Consortium and say, hey, there's one that's misreported here. We need to correct that. So there's more information here. There's a white paper, and I'm just going to jump out of the PowerPoint to show you on our website. So I just went to the DNASTAR website. Under Support, NGS Support Materials, where it says #1 in NGS Accuracy, there's more detailed information then about how we use that genome in a bottle data to run it through our pipeline to measure our accuracy. And there's a number of papers where SeqMan Engine is compared against GATK, which is kind of the open source standard for variant calling, as well as some commercial competitors like CLC and Genius. And you can see that these, I'll just click on one of these. We won't go through this whole thing right now, but it's a detailed background, and really the intent here is to give you enough information about where the data came from, where you can download the data that was used as input data and as the reference. Here's all the 3 exomes that we'll mention from the Garvan Institute and Arup and ICON, Mount Sinai, where we can download the actual data and run it through our software and come up with these same numbers. So again, it's reproducible. It's not just a marketing piece where we make claims. We provide data and all the background information for you to reproduce the results. And we'll look at these in detail in the slide. So again, there's white papers then that really go through that in detail. Okay. Oops. So DNASTAR provides this kind of validation workflow. Right now it's embedded in our gene panel workflows, and we call it our validation workflow. So it allows us, and I'll show you how we set this up, and it's this validation workflow then that allows us to determine what our accuracy is. And there's kind of a flow chart here that's I know it looks quite complicated, but I'll try to explain what we have. And this kind of upper branch here, there's a targeted regions file that we intersect with our answer. So this can be done different ways. You may have your own list of targeted genes in a bed file that you can use for your data set. For this particular data set, we took the NIST high confidence regions. Maybe I can highlight that. So I'm talking right in this area. So this we downloaded from Genome in a Bottle Consortium. So these are the areas in the human genome that are high confidence, that they can make variant calls to that very highest degree of confidence. There are some regions in the human genome where that's not the case. Some low information areas, for instance, where the alignments aren't as accurate. So this bed file then is, we'll call it 80% of the human genome, something like that. And then they also have a list of these several million variants that are called in that human genome. So we can take these files then and intersect them with a smaller, so maybe you're only interested in 20 genes. Well, those 20 genes probably intersect with the NIST high confidence areas. So we can make an intersection file to say, if I sequence NA12878 and I use my own bed file compared to these high confidence areas, there should be a set of Snips, variants that I detect every time. And I can measure that then and see how accurate my process is for the genes that I'm interested in. And this is a little piece that we can help customers with. but it is using these purple reference materials that I have in the flow chart essentially as the answer, and we can compare them then with your own bed files or VCF files to come up with an accuracy estimation. So again, we can help with this part, but so it's a multi-step piece. So the data then, the NA12878, goes through our assembler, SeqMan engine, which has a variant caller. The variant caller then produces a set of output files, and we can make that comparison in our software automatically to see, did we find all the variations that were in these reference data sets? Again, this is all kind of automated. We get accuracy calculations, and at the end, just a report then that just says, you know, how accurate were we? Did we find 99.97? We find 99.7, 100% in some cases. So it's a really meaningful and useful piece of software for these kind of workflows. So the way that it looks, and I'll show you in the software as well, is when we set up our assemblies, we have a wizard that makes it as easy as is possible to set up your data. I can assemble on the cloud as well. So if I want to run my assembly on Amazon hardware, I just log in, and everything else will look the same essentially. It's just that when I say go at the end, it'll just use that cloud computer. And here's the cloud data drive. So I'm using, in this case, the Garvon exome data. And here's some FASTQ files that can get compressed, get sent up to the cloud and assembled. So we load the data in, FASTQ input. Then we load the reference and the bed files. The reference files typically are downloaded from DNASTAR. So you can see these available data packages. We call these genome template packages, and they're available for quite a few of the different model organisms. And what they contain is the annotated reference chromosomes plus the dbSNP database files. So if your organism has a dbSNP database file, we can build a package like this. There are some organisms where that's not the case. The SNPs aren't curated at dbSNP, so we typically do not build genome templates for those organisms. In that case, we could use a reference file plus a VCF file, for instance, that's separate. So you can download these packages. In SeqMan Engine, you load the whole genome template package. Again, here's a human genome template package that's been loaded. And we can load the bed file or manifest file. And this tells us the region of interest. So if it's an exome, you're going to have all the targeted exons for that genome, or it could be a smaller panel, or it's just a relative handful of genes. So so here's kind of another way to show how we can use those high confidence areas from the Genome in a Bottle Consortium and find where they overlap, say, a gene panel. So this top range right here, the gene panel bed, you can see there's just a few little green tick marks. So it's a few, for example, a few genes that were targeted on this chromosome. Then we have the NIST bed file, which is about 90% of the chromosome, represented by the red line. And then the little purple marks are Snips that have been verified by Genome in a Bottle, by that consortium. And so we can find those areas where there's overlap between my bed file and the reference data set. And that's where we can help build that and make that confidence area. And then we can make an accuracy call with any data set that's using NA12878. Again, the sequence files, once we're loaded in on ground software, we can see that there's experiment names here, NA12878. I can run multiple experiments and have multiple samples in one setup. So you might have 50 exomes or 50 projects that you want to set up at one time. We just set a different name for them, and you just kind of click through the wizard. The VCF file contains these confirmed variant positions. So again, we set up the experiment, we pick the VCF file that contains all the answers, essentially. It's the answer key for all the SNPs in that data set. And by doing all this, we actually can measure different points in the process where you can actually lose some accuracy. And so this is what we'll look at in the software, because this is some of the stuff that you can't do this in a lot of other softwares. The other softwares do not give you access to the intermediate steps. You essentially run an assembly, you get a VCF file, but you don't get anything in the middle to really evaluate and interrogate where you might have loss in your accuracy. So one thing is coverage of target regions. When you do a targeting experiment, an exome or a gene panel, that targeting is very rarely 100%. There are almost always some missed areas. In exomes, it's typically about 5, maybe as much as 10% of the areas you intended to sequence are missed. So you need to have reports that allow you to evaluate how effective the targeting was across all your capture regions. There's also specificity of the targeted areas. Depending on the technique that was used to capture those areas, some are more or less specific. So a common issue in eukaryotes is that the capture technique actually grabs pseudogenes. And so you end up sequencing pseudogenes rather than what you intended to capture. And so you want to be able to assess that. Then there's the actual alignment algorithm. How efficient is the mapping? And how accurate is the actual alignment of the sequence data to the reference? And there's some variability there. We've got a really fast, accurate aligner, but GATK is also a fast, accurate aligner as well. So a lot of the differences then you're going to see come further downstream, and that's things like variant filtering. How effective can you filter out the noise from the true signal? And if you're too aggressive with the filtering, you start to lose true positives. So there's a fine balance there between setting the filters to keep the false positives as low as possible without sacrificing true positives. And there's always some loss. There's no data sets or softwares that will give you 0 false positives with 100% true positives. There's always some slight trade-off. So we give some access there. And all of these things combine then to the overall variant call accuracy in your samples. And so you want to be able to assess all of them in detail. There's also some interactive reports. We'll look at those in the software and the specificity. So we'll look at that. The metrics that are generated. So in the validation, we get a measurement of the true positives, the false positives, the true negatives, which is really important in a more clinical type setting. You don't want to miss variants that are there. So that's one of the most critical measurements is the true negative detection rate or non-detection rate. And false negatives are actually the most troublesome. So if you have variants that are disease-causing, for instance, you don't want to miss those. So you want to have your FN false negative rate as low as possible. True negatives, I was mistaken. There's lots of true negatives. That's going to be a very large number. It is the false negatives that you want to minimize as much as is possible. All of these then, and you can see there's some formulas, lead to false discovery detection rate, FDR, and then we use true positives and false negatives and true positives for sensitivity and specificity. So this is how we derive the different numbers, and then now we can look at some results in the table here. So we took this genome in a bottle data set, ran it through our assembler, our Snip caller, and then ran the statistical measurements for the accuracy. And over a period of several months, we compared it first to what we consider the standard in the industry. That's the BWA mapper and the GATK variant caller, which has 2 versions, the unified genotyper and the haplotype base caller. So we picked both because we know people use both, and they actually produce different results. So we thought it was important to do that. Then subsequently, we made comparisons to 2 of the commercial competitors that you might see the most, CLC Bio and Genius. And so this is kind of the last report that we ran, where it's really a summary then of many months of work with accuracy. And you can see the sensitivity of our software is tops amongst all the other softwares. So we have the highest sensitivity. The FDR, false discovery rate, we can see that BWA is actually a little bit lower than us. They have us just by a couple of tenths in some cases. They have a little bit low false discovery rate. The commercial competitors, though, have a very high FDR, so they have a very high number of false positives in their Snip callers. We get a number then of the number of true positives, and then you can see the number of false positives. So there's about 15,000 true positives in this data set. So this is, again, the NA12878 exome, which has been cross-referenced to the genome in a bottle answer. And you can see that the false positive rate for DNASTAR and for BWA, it's 200 or less. Commercial competitors, over 1,200. So there definitely is an issue there with the false positives. If we get to the false negatives, that's where DNASTAR, again, really shines. We have the lowest false negative detection rate, much lower than BWA and much, much lower than the commercial competitors. And then overall, the time that's elapsed, ours is at least twice as fast as the next nearest competitor, which would be CLC. So we think that with these results, we're pretty confident in claiming that we are not only the fastest, but the most accurate assembler and SNP caller that's available. So again, this is human exome NA12878. This one's from I believe, the Garvan Institute. We also ran this on additional exomes, just to see how consistent the result is, because different capture techniques have slightly different error profiles and depths of coverages. So we did the same thing with the Garvan Institute, the Arup Labs, and the Mount Sinai exome. You can see the Mount Sinai exome is actually a much bigger capture. There's 35,000 true positives captured, so it's a bigger data set. And you can see again that the FDRs, again, are in that low 2% range. Sensitivity ranges from 99.9 to 99.7, and about 2 hours. These other exomes are bigger, so close to 3 hours for the Mount Sinai. So we're consistent across exomes. We'll continue to test against other data sets as they're available from the Genome in a Bottle Consortium. So you'll see more and more data sets get added, but it looks very promising that we're producing consistently the lowest false negative detection rate, the highest sensitivity at the fastest time. One other thing I'm just going to add before we go into the software, we have a release coming out early in 2016 that will introduce human variant annotation database, which will pull in some information for helping to evaluate our SNP calls, so things like ClinVar and 1,000 genomes, in addition to dvSNP. So this is some new functionality that we'll have, and I assume that we'll have early next year, we'll have another webinar that covers some of these topics. So let me jump out of this PowerPoint. And I'm going to start with SeqMan Engine. And back up here to the start. So SeqMan Engine, again, is a project setup wizard. And the wizard is just letting me start with a new project. It'll guide us through the project setup. So I can pick things like exome assembly. For a validation workflow, it's going to have a control. So we just pick control. We name the project. Pick the disk that we want to use for our temporary files. Again, for anything aligned to a human genome template package, we want a 1 terabyte disk dedicated to handling those intermediate files. Those files get erased at each time an assembly is completed, so it just kind of is just a space that's used, kind of a scratch disk for those files. So I load in a human genome template package that I've downloaded from, there's a link here in the wizard. I download that from the DNASTAR website. The targeted regions file, so I pick the NIST validated regions. It's their bed file that has all the regions in the human genome that have been highly validated, so I've loaded that in. So you can see it's a pretty simple setup. On this page, we load in the sequence file. So I pick the platform, whether it's multiple sample or single sample, and then I just add the FASTQ data. So here's data from the NA12878 genome. I can load those FASTQ files, and you can see they're multiple gigs in size, so they're sizable data sets. Load those in. If I have multiple experiments, I can set them and name them. So I could have experiment A, experiment B, experiment C. So it allows me to set up multiple projects. And now, because I have a control, I can define what the control is. So I say, yes, this is a control. It's a validation control. There's a pull down here for baseline or none. And then I pick the VCF file that's my answer file that I want to compare to. So I can set the VCF file. Again, it's the NIST validated Snips. Load that in. And just some basic alignment parameters. We'll just stick with everything default here for a validation workflow. And it's ready to go. So we get this script that's written in the back end. I click Assemble, the assembler will start, and then I can load this project in to SeqMan Pro to look at the aligned data. If I want to run the calculations, we can do that right in ArrayStar. There'll be two buttons here that allow us to do that when the assembly is complete. And so if I say, show me the accuracy report, I'll kind of get this report. Again, ArrayStar does all the math here, and it tells me things like the number of true negatives, the number of false negatives. You can see there's 51 in this sample, false discovery rates, sensitivity and specificity. Right, so those are different values and balanced accuracy. And then I get a little bit more detail and to see what my accuracy, so a real common question we'll get is how deep do I have to sequence my samples to get the best accuracy? You know, do I need it to be 1000x, 100x? Is 10x enough? And so we actually calculate the accuracies at, at different depths. And we can see, usually about 20x coverage our sensitivity is optimal. You can see even at 10x we can be pretty close though. So FDR goes higher as you get thinner coverage, and it seems to be optimal. Right around 20x we get really nice results. Of course there's a trade-off with positives. Sometimes you have thinner areas of coverage where you have some SNPs you're interested in. So if you're tempted to go as thin as possible to pick all those up. So if I go even to 1x coverage, I'm finding 16,000 true positives. But if I do 100x coverage, there's only 9,000 SNPs I can detect at 100x coverage. So there's this kind of trade-off between how deep do I need to sequence before I'm confident. So this kind of a report really helps out with that. So again, this is the validation report that just tells us for that data set how accurate is my SNP calling. If you see something that doesn't, imagine if the accuracy isn't as high as you want to go interrogate. Well, why isn't my accuracy 90, above 97%? If it's a little bit lower, we have tools that allow you to go in, and SeqMan is one of those tools then that allows me to open up the assembled data. So here's the NA12878 assembly. Here's chromosome 1, 2, 3, 4. I can highlight this header and generate a report for the whole genome. So one place I would go is the coverage of the targeted region for the whole genome. And so that's under the project Show Coverage of Targeted Regions. It takes about 10 minutes. This is a really big report, so it takes some time to build this whole thing. So I did it ahead of time. And what it is it's just reading the bed file. In this case, it's from an exome. So each one of these rows is a different targeted area in my exome. And I can see the chromosome, so this is all chromosome 1, the feature, the location, the length of that targeted area, the depth of coverage, the percent of that targeted area that was covered, a normalized depth value, an RPKM, number of reads. And I can see here, there's one right here that is not fully covered, and I can sort by coverage as well. Or I can just kind of scroll through and look, here's one at 81.3. I can double click, go to that point in the assembly, so it's all interactive. And I can see there's a thin area here, so the sequences were captured, but not over the entire targeted area. Right, so I can look at that in detail. There I sorted by percent covered. And I can see, again, this is an exome. Here's all the targeted areas that have 0 coverage. And I'm just going to scroll down, so keep an eye on when we start getting covered areas. So I'm scrolling down. There I've got coverage, and right about here I'm at 100. So if you look at my scroll bar, you can see, it's about 90% of those targeted areas, the low 90s, have 100% coverage. But it is, in an exome, it's very common to have this number of regions that don't have any coverage or have minimal coverage. So if that's a gene of your interest, you're going to want to know that right away. Look at the report and say, well, either I have to kind of redesign my capture, or maybe I design, and do Sanger sequencing if it's a really important area. So you need to have this report quickly to assess whether or not your targeting was effective enough. So it's a great kind of a report for that. There's also a tab up here, Not Targeted. Here are sequence reads that align to areas outside of the intended regions. And when you open the report up, one thing that really jumps out is that the features here. there's a bunch of pseudogenes here. And you can see there's significant depth for some of these too. If I double click, this is all in a pseudogene. I can twist the triangle here. So I can see that this capture technique, there was quite a bit of sequence data that went outside of that area. Again, it's exome scale, so overall, it's not a problem, but it's something that you want to be aware of and be able to, again, interrogate and see how much of my data went outside those targeted areas. You can sort here by depth, or I can see, 2,000x deep. So there's a lot of pseudogenes. So this is just kind of the noise that's typical in an exome, but you really want to take a look at it if you can. There's also a summary of the effectiveness of the targeting. It's called the Enrichment Report, and it just gives you an idea of the total length of the targeted regions. So this exome, that is, let me see, that's 27 million bases is what's targeted in this exome, and then you get a numerical count for how many reads aligned and how many are aligned in that targeted area, and you get this kind of an enrichment, and what percent of the target was covered at 10x, 93.29 in this case, how much at 20x, 91, how much at 50, 81. So again, it's a good summary then of that targeting, and really critical information when you're doing this sort of work. So that's the targeting. There's also actually looking at the SNP calls. And so this is a SNP report then across the exome, and there's all sorts of information in here. I won't go through all of this right now, but there is things like reference position, the type, whether it's a SNP or an indel, the reference base, the genotype call, if it's in a splice site or not, if it's in a homopolymer, the SNP percent, the p-value, the p-naught ref, whether or not it's in the region capture. By default, we filter to only show SNPs that are in that capture. The DB SNP ID, cosmic score, GERP, evolutionary rate profile. The user ID. The user ID is the number that's in the VCF file. So this is the genome in a bottle database number. So I can say they all have a user ID, or most of them. So those are the SNPs that were in my answer, essentially, my answer that I compared against. And then I get protein changes. I can control what's here. I could say, well, I don't really need contig position, and I don't need the, maybe I'm not doing cosmic or Q-call or homopolymer. So I can edit this and make it more conducive for my analysis. Or I can apply more filters. I can say, show me just those SNPs that are in that VCF database. And there I can see I've got 16,710 SNPs that were in the genome in a bottle database, and now I can go and look at them. This is all interactive, so I can double click, go right to that point. And this is where SeqMan is really fantastic, is that I can go to that point in my assembly, look at the annotations, look at the aligned data, and see the SNPs clearly called there in blue. So of course I'm not going to look at 16,000 SNPs. I'm going to trust that those are accurate calls. I might be more interested in this other tab, which we call the missing SNPs. And what that means is these are SNPs that we were supposed to detect these. They have a user ID here, and there's 44 of them. So these are the ones that we missed. And so this is where our focus is now, is we found 16,000, but what happened to these 44? And so this report allows me to go and look at them. So you can see right away that one thing under SNP percent, a lot of the SNP percentages you can see are right around that 20% threshold. So for a heterozygous call, that's a pretty weak heterozygous call in many cases. And so we can start to look at them and just kind of see, is there any reason why we would have missed a SNP there other than it just didn't occur frequently enough. And here you can see it's a 20% call, so its p-value is a little bit too low. And you can so this is a nice way to kind of go through, and we do this from a development side. We say, well, is there any way we could have detected that SNP? Is there anything that we could have aligned better here? Now, in these cases, the alignments all look good, so I don't see anything there. And this is how you or maybe the SNP, so here's what I was kind of looking for. So here's an area you see some gaps, and we'd look at that and say, well, the SNP was supposed to be at one location, but looks like it was right next door. So maybe it's a little more complex area here for a little repetitive element that didn't get aligned quite efficiently. So we can kind of look at these areas and say, well, we could improve the alignment. Maybe we can't. Maybe that's what it is, or maybe the SNP really should have been next door, and we go back to the database and say, well, we think the SNP actually is one base pair left or right of where it was listed in the database. So again, it's only 44, so but it is worth looking at them just to get a feel for what's getting missed. So those are some of the tools then that we provide. Again, it's an easy way to set up an assembly in SeqMan Engine. You can pick a control. If you don't want to use the genome in a bottle, you may have your own data set where you say, I've got 100 SNPs. I'm going to have my own VCF file, see if DNASTAR software can align and detect all 100. That's another way to do this as well. The genome in a bottle is nice because it's over 10,000, over 15,000 variant calls. So we set up the project, we give tools for analyzing the project, we generate an accuracy report. So it's really a nice workflow. Now just to finish up here, some things I know that many of you are interested in what we're doing in the future. We have the variant annotation database that will pull in some clinical type information into the analysis. Another focus for version 13 are new RNA-seq tools, and we're going to be streamlining those RNA-seq workflows. We'll see some additional white papers with RNA-seq in the next few months. And again, as I mentioned earlier, just the enhanced data analysis and database utilization. We'll continue to work in those directions. So with that, I'd like to wrap things up. If you have any questions at all, I'll stick around and answer any questions that come up. So again, thank you.
Thank you, Matt. That was an excellent presentation. We do have a few questions. I'll start with the ones we have, but
okay.
Just as a reminder, people can continue to chat questions in over the next several minutes. So you kind of mentioned briefly that you can validate your own gene panel or exome data sets. Can you talk a little bit more about how you would do that? The focus today is kind of more on these standard data sets.
Right. So really the key there is that if you have a data set, say, most commonly it's data that people have sequenced with Sanger data in the past, so it might be a handful of genes, and so if you know that those variations are present and have validated them yourself, what you can do is create your own bed file and VCF file. And just like we did for Genome in a Bottle, where we had the bed file from Genome in a Bottle and we used their VCF file, you can create your own from an Excel spreadsheet and run it through the same validation process. And that's something we can help customers with if you need to tweak the format a little bit to get it from the Excel to the bed. And we also have, in our help menus, we show specifically how to build those files, and then you can validate using any data set. The key really is to get a data set that has answers that are 100% accurate. If your answers aren't 100% accurate, then your accuracy will be, less than 100%. So.
And just as a follow-up to that, so that you can do this kind of validation with non-human data sets as well? Yes.
So it can be any kind of, any kind of a system. It doesn't matter. It just comes down to do you know where the variants are supposed to occur? If you know that information, you can create a VCF file, and we can run a validation assembly from that file.
Great, thank you. And just one last question at this point. I'm not seeing more come in, but we do have a few minutes. Is there a way to look at some of these validation metrics by the specific variant type? So, for example, SNVs versus insertions and deletions. Is there a way to parse some of that information out?
So we don't parse that out specifically into different reports. So the way that you would do that is create a separate VCF file that contains only substitutions or only indels. The validation that we run that I showed you today includes both. So it is Snips plus, indels up to probably 15 base pairs in size. And so if you wanted to know how accurate are we just detecting indels that are 5 to 10 base pairs, what you would do is create from your VCF file, you just have a VCF file that contains those variations. The challenge there, of course, is that indels are rare, so there's a much lower number of indels in that size range, across even a human exome. So the number of true positives that you have is going to be a much lower number, but yeah, it can be done that way.
Great, thank you. Just as we wrap up here, I'm going to switch back to my desktop and show you a couple resources, some of which Matt mentioned. I believe you should be seeing my home screen now. Here we have the DNASTAR home page on www.dnastar.com. And just want to point out a couple of things. Matt mentioned our accuracy papers. That's under our support menu. So again, if you want to look at any of those white papers in more detail, that information is all available right here. And then additionally, from the home page, you can get to our webinars page. And we have another webinar coming up next month. I'll be presenting on Sanger sequence analysis tools. Matt mentioned briefly some of our Sanger analysis workflows, perhaps something you're doing to build some of your data sets for validation. So if you're interested, please sign up for that webinar on November 18th. And then also, the recording of today's webinar will be available on the same page under the Next Gen Sequencing tab. And here you'll also find several other webinars that talk about some specific workflows in more detail, an overview of our genomics suite, for example, gene panel workflows, and many more. And then just finally, Matt also mentioned our videos. Those are another great resource if you're just getting started with our software. We've got several short videos under our Next Gen Workflows category. So you can look at, maybe the specific data type you're working with, or various workflows and analysis options. Just get little 2 to 3 minute snippets of how to get started and get things set up. And then, of course, we're always here to help. So if you have questions about today's webinar or our software, feel free to contact us at webinars@dnastar.com. And again, if we didn't get to any of your questions today, there were a couple that came in that were a little more technical in nature. So we'll be sure to follow up with you after the webinar. So we thank you all for joining us today. We hope you found this webinar informative, and we hope to see you again at another webinar in the future.